Dallas Hartwig is a functional medicine practitioner, licensed physical therapist, and author of the New York Times best-selling book, It Starts With Food, with his lovely wife, Melissa, which she just walked out with their beautiful little boy. He acts as the chief eating officer, CEO, huh? That's another version. Hey, okay, maybe I get to do that. At Whole Nine, and describes himself as a compulsive learner and an avid snowboarder and motorcyclist. Dr. Helen Messier is from Vancouver, Canada. She holds a PhD in molecular genetics and immunology and an MD in family medicine with certification in functional and integrative medicine. She is passionate about using the power of genetics to optimize each individual's needs. Please welcome to the stage Dr. Helen Messier and Dallas Hartwig.
Thank you. Thank you. I would say,
yeah.
Okay.
So thank you all for coming. We're really excited to be here and to kind of talk about a topic that is very near and dear to our hearts, near and dear to Helen's education and clinical experience, and becoming more and more near and dear to me as I understand the clinical relevance of some of this stuff, and I'm certainly much newer to these topics than other people like Helen. So we want to talk a little bit about this concept of genetics, personal genomics, and what that means in terms of influencing your life. People often think about genetics as these things that are built into us, these things that are influencing who we are and what we become and how we interact with the world around us. And we think about things like eye color and things that are very sort of predetermined. But the point that we really want to get across with a lot of this discussion is that while genetics are profoundly influential in who you become and how you act and how you respond to the world around you, it's not a predestination sort of concept where your path is laid out for you and you just kind of got dealt a really bad hand. So we want to kind of break down some of that complexity, understand the influence of some of this in terms of things that are modifiable and how you can make more educated choices in your day-to-day life. So how many of you here are truly healthy? We've got a few takers. Brave, I appreciate that. How do you know?
The question is, how do you know? And I'm just going to excuse my voice. I have a really bad cold, which is why I'm hooked up to the mic, but I'm pushing most of this over to Dallas because I can't talk very well today. But yeah, the question is, how do you know? Anyone brave enough to say why they're healthy or how they know? Good shape.
So yeah, so the concept of being physically fit and being kind of physically capable as an indicator of health overall, I think that's a good piece of it. Kind of our view on what health is a little more kind of multifactorial, and it's certainly more than the lab tests, the metrics that you can get by going to your primary care physician's office and doing blood work and looking at things like total blood cholesterol, HDL levels, LDL levels, what your body weight is. Are you know, people like, well, I'm relatively healthy body weight. My cholesterol's good, so I must be solid. I must be super healthy. Body mass index is another kind of correlate with body composition. Blood pressure. Everyone knows that it's not healthy to have really high blood pressure, so everyone thinks that if your blood pressure is good, you're totally solid. Fasting blood sugar. Body fat percentage. All these things are sort of numbers that we can measure and give us sometimes a false sense of security whether we are truly robustly healthy. And I think to your point that there are some sort of more subjective and experiential things that contribute to kind of your overall health status. And it's much, much more than just, can I do this thing? What does my number say? What does a number in the scale say? So our perspective...
Yeah, I mean, I like to think a lot of people, so this is a different crowd. You know, you're looking at what you can do. Most people say health is the absence of disease. Well, we know it's a lot more than that. And I like to think of it as optimal health as a state of no limitations, where you can do anything physically, emotionally, mentally that you want to do. If you want to climb that mountain, if you want to, you know, give that talk, if you want to run the marathon, you should be able to do that. And there shouldn't be anything limiting you from it. So it's a state of no limitations. Often I like to think of health, it's on a continuum where optimal wellness is on one side of that continuum, and that's really where we all should be starting from. You know, if we all have a good prenatal environment in birth, we're starting from a point of optimal wellness without any of those limitations. But over time, what happens is we don't always live a lifestyle that's in tune with our genetics, and we'll get into that a little bit more. But we have stress, we don't eat properly, we don't exercise, and we start developing these physiological imbalances. Those are things like oxidative stress or inflammation, things that start happening in our body that we don't really know that they're there unless we specifically look for them. And if we don't look for them or do something about them, over time we start to get dysfunction. Dysfunction is where we get symptoms. So symptoms like we get a headache, or we get a bit of fatigue, or a little bit of abdominal weight gain. And this is where I would put those numbers, right? Not a healthy blood pressure, or cholesterol numbers are off, or a body mass index is off. That's in that area of dysfunction. Those physiological imbalances have already progressed to something we can identify. If we don't do anything about that dysfunction, over time we end up with disease. And disease is just an arbitrary point along this continuum. And then we end up getting disintegration and death. Now, obviously, up until disintegration and death, this is all reversible. We can all get back to that point of optimal wellness. So I like to think of really, this is a functional medicine analogy, but think of an analogy of a tree. We're all like this tree, where what we see is the leaves, right? And our leaves can be robust and green and healthy and look really good, or our leaves can be yellow and shriveling and starting to fall off. Now, in our typical Western medicine world, what we do when we see a yellow leaf is we take some green paint and we paint over it. And then, you know, it looks good for a while and we feel good for a while, but we're not really getting at we forget that we're connected by a trunk and we're connected by roots, and that we all live in a soil. We all live in an environment. So the trunk is the physiological imbalances. Those are the things, the body processes that have to happen in every cell of our body and in our entire body to keep us functioning and healthy and keep those leaves green. But that the roots of the tree, those are our genes. And those genes act very closely to the soil that they're in. So the soil, being our environment, our lifestyle, the food we eat, the water we drink, the thoughts we think, the stress we're under, the sleep we have, they get filtered through those roots up into the tree, resulting in physiological imbalances or not. So the genes are the roots. And what we have to remember is that not all trees need the same environment. We all have different roots. So take a look at the person next to you. Uh-oh. Did I just do something? Oh, Adobe update. Not now.
We really, really strongly believe in being current with your software.
Exactly. So, a pine tree needs a different soil, a different environment than a palm tree or a maple tree. And so we all need a different environment that's going to act with our genetics to result in that healthy tree or not. And so I was just having everybody look at the person next to you. Do you look the same? No. You probably have a different eye color, different skin color, different, exactly. We're not the same, and so we don't need the exact same soil to grow in. Our soil needs to be slightly different based on what our individual genes are. So
then in order to optimize this, to optimize our health, we have to take that context, that soil and root context, and say, we need to understand where we come from and what we're built from, and that background. And we're really talking about that genetic background. And for those of us here kind of understanding and learning and excited about that paleo concept, that ancestral health concept, it's not really, we already kind of are thinking about the idea that our genetic makeup really profoundly impacts who we become, impacts our health, and really frames what our behaviors could or should be to reach that place of optimal health. We we're familiar with the concept of a mismatch of genetic predisposition or genetic heritage that we then put into an environment, and we put into a soil environment that's totally different. So if we take a maple tree and we plant it on a beach in Tahiti, it's no wonder that the maple tree is not going to be optimally healthy, because that's not the right environment for that set of genes. You with me? Okay.
Yeah.
So then we have to understand where we come from, what that genetic heritage is, so that we can create and make the best possible choices of what that soil environment is in each of our lives. And it's different for each of our lives, just like the person sitting next to you looks different.
Exactly.
So then we have that foundation. We also have to identify some of those clinical imbalances, and that hopefully is the place earlier in the process, before the leaves start to turn yellow and fall off and the tree starts to disintegrate and potentially die.
Right. So, I mean, one of my mentors, Dr. Jeffrey Bland, had this quote, is that our health and disease patterns are not hardwired by our genes, but are a consequence of the interaction of our genetic uniqueness with environmental factors. And we're found, we fall right in the middle, and that's us. So we need to understand our genes, and we need to understand our environment and find the right match between the two of them. So the question is, how many of you are living in harmony with your genes? No hands going up this time.
No takers this time. We scared everyone off.
And that is the point, is how do you know? And that's what we want to talk about, is that we're at a state with science today that we can start looking for that and start understanding how we can live in harmony with our genes, our individual genes. Because from an ancestral, the ancestral movement is about living in harmony with our genes. It's about living a lifestyle that humans were supposed to live. But we're not getting to the level yet of that individual customization of our environment to our genes.
So when we say genetics, there may not be a really clear picture of what that is. So we're going to kind of get a little bit into what that means a little more, and we'll kind of breeze past some of the really dense physiology stuff. But we want to kind of give you a primer on that to frame discussions of what this can look like for each individual person later on.
Yeah. So we're going to talk about a little bit of genetics 101. I don't know if there's volume here.
That's all right.
Can you hear that? So inside our blood vessels, we have red blood cells and white blood cells. And inside the white blood cells lives our DNA. And it's wound up very tightly when you unwind it. So this video was made by Illumina, and I'm using that with their permission. Illumina is a genetic sequencing company. They build the machines to actually do the genetic sequencing. So let's look at this DNA a little bit. So DNA is the molecule of our heredity. So when we talk about that, and I know a lot of this will be review, but it just puts it in context of what we're going to talk about later. So in the center of every single cell of our body, we have this double helix molecule that we call DNA. And DNA is made up of a sequence of letters, or what we call base pairs. So on every strand, both strands, we have these base pairs that match up with each other. And they match up very specifically in that an A always matches with a T, or an adenine always matches with a thymine, and a G always matches with a C. That happens every single time. And it's the sequence of these letters, just like the sequence of letters in our language, that gives us the information. That's what codes for the information that our DNA has. So genes, actually, the DNA codes for something called RNA, which is the intermediate thing that takes it from the RNA, then codes for proteins. So each 3 base pairs is called a codon, and 3 base pairs codes for 1 amino acid. That's how the language works. That's how it's translated into proteins. And so it's these sequences of amino acids that get together and create all the complex proteins that we have in our body. Now, a gene is really the unit of DNA that codes for one or sometimes a set of very closely related proteins. And a gene can be from 1,000 base pairs long, or it can be over 1 million base pairs long in humans. So they're very variable. So proteins, they're the workhorses of our bodies. DNA is the information, it's the software, the proteins are the hardware. Proteins can be structural, so they make up our muscles, our bones, our cell membranes, or proteins are also enzymes, and enzymes control every single biochemical reaction that happens in our cells and in our bodies. And because we all have different changes in these genes, that results in different proteins in all of us, and that's what makes us unique. So how much DNA do we actually have? We have about 3 billion base pairs. That's a lot of base pairs of information, and we have this in every single cell of our body. So in the nucleus of every cell are 3 billion base pairs. If you took the DNA from one cell and you stretched it out, so it's lying along in a line, it'll be about 3 meters long from one single cell.
That's about 10 feet for you Americans.
Sorry, yeah, that's the Canadian version. And so because we have about 10 trillion cells in our body, if we took the DNA from one person and we stretched it out end to end, it would reach to the sun, from the Earth to the sun and back 100 times. That's from one person. So that's a lot of information that we have. In order to get all of that DNA into a cell, which is, as you know, microscopic, it has to be very tightly packed. And that's what you saw in that video, how it's wound up very tight into these little packages we call chromosomes, which are these little things there. So it was interesting that the human genome's only been sequenced. We only know the full sequence of it for about the last 10 years. That's when the Human Genome Project was completed. And at the time, we actually knew that fruit flies, or Drosophila, we knew they had 4 chromosomes, and we knew they had about 15,000 genes. And so we thought that humans would have way more genes than that, because we're obviously a lot more complex than fruit flies are. However, when we finally sequenced it and looked at how many genes humans had, we only had about 25,000 genes, which, if in comparison to Drosophila or fruit flies, that's not a lot more. So, you know, our DNA actually, the genome, we only about 1/60th, that's 1/60 of our genome, actually codes directly for proteins. The rest of that DNA, which at one point we used to call junk DNA, we know it's no longer junk DNA, actually directs when and where each gene is going to be used. And that's what makes us so complex. So that's why we can have 98.8% of our DNA the same as a chimp. Because, and I was got these a little backwards, but in each individual, we're only 0.1% different. So between each of us, we're 99.9% the same. But that variation that we're talking about that makes us unique, if you take 3 billion base pairs and you take 0.1% of that, that results in millions of differences between us. And therefore, that's why we're 98.8% the same as a chimp. So let's just take a little closer look at this 0.1% difference that makes us unique. What's it like? I like analogies, as you can probably tell already. And there's the analogy, I always use the analogy of an encyclopedia. So you remember those Encyclopedia Britannicas that we had. I'm trying to find a good technological, digital age analogy, but the encyclopedia still works best.
I feel like in 10 years, that analogy is just not going to work anymore. No one's going to remember what an encyclopedia actually is.
But for now, I think everyone's nodding, so I think we're good. So this is our great human genome encyclopedia. We inherit 23 volumes from our mom, and we inherit 23 volumes from our dad. Those volumes are identical, almost. Okay, they're identical, but we're going to go over how they're subtly different. Okay, so that makes 46 volumes altogether, and this is what those volumes actually look like. That's human chromosomes. There's 46 of them in every single cell, and it's the exact same information in every cell. So a brain cell, a heart cell, a muscle cell, a toe cell, we all have the exact same DNA in all of those cells. So let's look at the first order of difference, the first order of variation, something called SNPs. So if we take, say, volume 6, paragraph 3, sentence number 2, we can see that Dad was the center of attention. And if we take that exact same spot from Mom's volume, we see Dad was the center of attention. Now, I don't know if that's big enough for you to read, but you can see that center in Dad's volume is spelt with an R-E, and center in Mom's volume is spelt with an E-R. That's what we call a single letter change or a single nucleotide polymorphism, a SNP. We've all heard the term SNP bandied around, but that's what it is. It's a single letter change that in this case doesn't change the meaning of the word or the sentence at all. It's the exact same meaning. Now, if you took a whole bunch of these single letter changes, we could now tell you whether this encyclopedia was spell checked with American spelling or with British spelling. And so you can tell from all these collection of SNPs in a person's genome where that genome is from, the ethnicity of that genome. In this case, it doesn't change the meaning, and we're going to get into an example where a single letter change can change the meaning of the word. But this is an example of a SNP. You can see that the GC on the top is changed to an AT in the bottom. Excuse me. Okay, here's another order of difference in the two encyclopedias, or the two versions of the encyclopedia. Dad is an amazing guy, comes from Dad's genome. In the exact same place, same spot in Mom's genome, we've got Dad was an okay guy. The general sense is the same. It doesn't change it too much, but it's slightly different, right? So it's just a little bit different. And this is an example of an allele difference, and you've probably heard the term allele before as well. An example of an allele would be the gene for eye color, for example. So that gene, whether it's from Mom or Dad's encyclopedia, they code for the protein that determines the crystalline structure of our eye, and a little bit of a change in that will result in how the light is reflected to whether you see blue or whether you see brown eyes. And this is an example of on the chromosome, same gene, slightly different variation. You can have either blue eyes, as on the right, or brown eyes, as in the middle, or one of each, as in the far left. And that would be, in that case, it would turn out to be brown eyes because brown is a dominant allele, so it would show up as brown eyes. The next variation or level of variation is something we call a mutation. And this is where a single letter change can completely change the meaning of a sentence. So here we have Dad is amazingly cool in Dad's encyclopedia. And in Mom's encyclopedia, Dad is an amazing fool.
The third genotype in this set is actually Dad is an amazing tool.
Exactly. And so in this case, a single letter change can completely change the meaning of the sentence. And in some cases, it can change the meaning of the whole paragraph or the whole chapter. Here's an example of a single letter change that results in a mutation. So this is a gene that codes for the beta subunit of the hemoglobin molecule. And you can see on the left, that's a normal hemoglobin molecule. And it might be a little bit too small, but you can see one little A is changed into a T right there, and it results in a molecule, or a hemoglobin molecule, that looks very flat and misshapen, which is what we call a sickle cell hemoglobin molecule. So just one single base pair change, a mutation, can lead to a profound change in the structure of the protein. Now here's a level of variation that we actually didn't even know existed until about 8 years ago. It was only discovered then. And if you look at, say, volume 23, chapter 4, paragraph 6, in Dad's volume, it says, this is the story of how Dad and Mom met. Dad was such a great guy, and Mom was just smitten, so she married him. And in Mom's genome, it says, and this is the story of how Dad and Mom met. She married him. So in this case, a whole section of the DNA is missing in Mom's volume. So this is what we call copy number variation. And we can have from thousands to hundreds of thousands of base pair sections that are either completely gone, missing from someone's DNA, or duplicated. We have 2 or 3 copies of that gene. And this is an example of, you can see in the middle, you have 2 copies of C, which is the blue one, and then on the left, you have a deletion. So one of the chromosomes doesn't have that copy. The gene is completely gone. And on the other side, you have 3 copies of that gene. Now, this becomes really important because we should have 2 genes. So the function of all of our genes is calibrated to 2. When you have either like 1 copy, less copies, or more copies, it completely changes the calibration and the functioning of the proteins that those genes encode for. And that becomes very significant clinically and in terms of, what we see in our bodies. So all of us have these copy number variations. Typically, we have about 15 of these very, very large sections of DNA that are either deleted or duplicated. And in some cases, it can be completely, not just 1 copy, but if you don't inherit a copy from mom or from dad, you have 0 copies of a gene. And we'll go over an example of this where there's genes where we actually are completely missing the gene, and it becomes very relevant. So let's take a closer look at some of these examples of these genetic variations in people to see how they affect our health. So do you want to do this one? Or I can do it? Okay. So one of them that you're all very familiar with, I think, is the MTHFR, or the methyl tetrahydrofolate reductase gene, which codes for the MTHFR enzyme. And we know that when there's a small change in that gene, it results in many, many health problems, from bipolar disorder, mood swings, depression, anxiety, autism, heart disease, stroke, cancer, miscarriages, macular degeneration, infertility, high blood pressure. There's just so many that are linked to a single change in that particular gene. Well, what does MTHFR actually do? MTHFR, as you can see down at the bottom there in the red, MTHFR is a gene that codes for the enzyme, and that enzyme is responsible for taking folic acid, either from supplements or from our diet, and converting it into methylfolate. Methylfolate is the active or usable form of folic acid. It's what our body needs to use. We can't use the folate that comes from our diet or from folic acid. It has to be converted before it can be used. People with a change in that enzyme, they can't do that conversion very efficiently. Sometimes, quite a big reduction in their ability to do that conversion. So they don't have enough of the methylfolate around. It's very important, and it results in all these defects because it's in the middle of many different biochemical cycles in our body. You can see that in the middle. So let's take a look at some of these things. First of all, methylfolate if you go to the left, we need methylfolate in order to make all of our neurotransmitters. We need it to make our serotonin and our dopamine. That all happens in our brain. Neurotransmitters, as you know, move the signal, our brain signal, our neurological signal. And if we don't have enough neurotransmitters, that's when we get addictive behaviors or the autism or the depression, all of the anxiety. Now, it's interesting, and this is a little bit of an aside, we have to get the methylfolate into our brain. There's a receptor called the cerebral folate receptor. That's the receptor that binds to the folate and brings it into our brain so that we can make those neurotransmitters. There is an antibody in a lot of people called the cerebral folate receptor antibody that will bind that receptor and prevent the folate from getting into our brain. The interesting thing is that there's a very strong cross-reactivity with that antibody and antibodies to dairy. So a lot of people that have reactions to dairy or make antibodies to the dairy proteins, they will cross-react, bind that cerebral folate receptor and not be able to get folate into the brain to make their neurotransmitters. You often see these guys that when they drink milk, they get depressed, or they just, have that brain fog that hits them when they drink dairy or consume dairy. You see that in kids that are breastfed that suddenly get switched to cow's milk and they start getting autistic type symptoms. That's one of the reasons for that. The other thing that we need methylfolate for is for our methylation cycle on the right. The purpose of our methylation cycle is to make SAMe, which is that one up there, S-adenosylmethionine. SAMe is our methyl donor. It's needed we have many, many methylation enzymes, and SAMe donates that methyl group for all of those reactions, more than 200 of those reactions in our body. And so it's as important, almost as ATP. We know that ATP is our energy source. It's our energy currency. If you don't have ATP, you die. If you don't have SAMe, you die. You need that to maintain our functioning. So if you block this cycle from turning, you also start to accumulate homocysteine, which is down here on that cycle. And we know that high homocysteine levels are linked to heart disease. They're linked to a lot of different health problems. And then homocysteine goes down to make glutathione. Glutathione is our master antioxidant. It's our master detoxifier. So when you block that cycle, you're preventing glutathione synthesis, you're increasing homocysteine again. That's why it's linked to so many different health problems. Another interesting tidbit is we also have a genetic change in this. Some people, there can have a genetic change in the enzyme that converts homocysteine down to cysteine. It's called the CBS enzyme. And the change in that enzyme actually upregulates it. It makes it work faster so that it'll suck the homocysteine down. So, and what the relevance of that is that homocysteine is not the perfect marker for looking at MTHFR status, because you may also have that second genetic change that will lower your homocysteine level, and you may still have the MTHFR problem. So it's a marker, but it's not a perfect marker. It's better to look at the genes. So if you have, let's just stop going forward here. I'll just do it manually. So if you have a SNP, or genetic change in one or more places of the MTHFR gene, your enzyme activity is reduced by 50 to up to 90%. So if you have that change, it's really important to supplement with methylfolate. Dietary folate, important, but it comes before that block. So you're not going to be able to compensate well enough by increasing just dietary folate. And now my voice is just about done, so you're on.
So that's one example of one SNP that can have profound effects on your health, certainly through mental health, through cardiovascular health, for essentially risk, kind of a risk mediator for so many different disease processes. And let's just talk about another kind of interesting SNP that affects our behavior. So we all know that food choices are a profound influence on our health overall. And for those of us who've kind of read about how food works to influence the signaling in our bodies, to let our bodies know that we're fed, we're good, we're healthy, we're strong, that concept of satiety is a really important idea, and that's one of the kind of driving factors in making consistently good food choices, where your food should be nutrient dense and should also provide you with adequate satiety signaling so that you know when you've actually eaten enough of this good, nutritious food and you can stop eating because you've eaten enough. So that satiety signaling needs to work really well for you to have a good, healthy, self-regulated, intuitive, natural relationship with your food intake, because that's a normal regulatory mechanism. And there's a SNP in a particular gene that influences one aspect of that satiety signaling. So the FTO gene and there's different genetic variations that are strongly correlated with body mass index. And one of the mechanisms, or the primary mechanism that happens, is it mediates our satiety signaling. So in the case of the FTO gene, it's expressed everywhere in the body. Remember that the DNA is the same in all the cells of our body, but it's especially expressed in our brain and liver, which of course are critical for that metabolic regulation. So in the hypothalamic nuclei, which is where a lot of the regulation of energy balance takes place, am I hungry, am I full, do I need to do some physical activity, am I just not really hungry at all that regulation takes place largely in the hypothalamic nuclei. And that's partly regulated by feeding and fasting. So that's where how much you eat and how often you eat will influence expression of some of these genes in your brain. So then there's a SNP within the FTO gene that has one particular variant. We call it the A variant, and it's strongly correlated with obesity. And in the kind of the media, they've kind of taken this and really kind of stretched it out to make it headline worthy, and they call it the fat gene. I don't know if you've read anything about this. And interestingly enough, if you look at different ethnicities have different prevalence of this A variant. So up to 46% of people from kind of Western European descent have this A variant, whereas only about 16% of people from a Chinese ethnic background have this same A variant. So ethnicity, i.e. our genetic heritage, strongly influences the likelihood that we do or do not have some of these variations. So then this A variant then influences actually the structure of our brain, like literally how big different parts of our brain are, I don't know. But the thing that I think is really interesting, if you look at MRI imaging of volume, literally of how big different parts of your brain are, the A variant carriers have smaller, an 80% smaller volume of the frontal lobe, and the frontal lobe is involved with executive function and decision making. And so if you have actually structural shrinkage of that area, it's not too much of a stretch to understand that decision making and executive function are related to that piece there. So you can see on the diagram on the far right, you can see the frontal and occipital areas of the brain that are most affected in terms of differing brain volumes.
Same thing there.
Yep, and the same thing here. So when you look at, actually go back one, when you look at brain imaging of people with higher or with different BMIs, you can see changes in those same brain areas. So we look at different FTO variants, and we see changes in brain volume. And then we look at people who are either obese or not obese, and we see the same changes in the same parts of the brain that are very tightly correlated with those genetic differences. So then with people with the A variant then compared to the T variant, which is the non-at-risk category, it didn't seem to change their energy expenditure or their VO2 max, their maximum oxygen consumption, but they consume significantly more calories. Right? So it's just the whole concept of regulating satiety. If you impair that satiety signaling and that off switch doesn't work very well, you're going to tend to eat a lot more food. And specifically, you're going to tend to eat a lot more food that's very calorie dense. So what this looks like then in terms of how much you enjoy food and how well that satiety signaling works, the A variant, so this is the group on the far right in both those graphs, the satiety responsiveness is basically how rapidly and how well do you respond to food when you eat it. And so what we see is the A variant on the right-hand side has less of a satiety signal in response to food, but the second graph there, they enjoy food so much more. So it kind of puts you at risk if you've got an endless supply of food around you in the modern world like we do, especially processed and sort of hyper-palatable food. You can understand then if you enjoy food so much more and you don't have that off switch, that satiety signaling that works as well, then it's going to predispose you to overconsume some of these energy-dense foods, thus contributing to some of the metabolic dysregulation and obesity that happens when you do that behavior.
Right.
So again, the FTO affects food preferences. They tend to crave those energy-dense foods because those are some of the things that are the ways to correct what your body perceives as an energy imbalance, because the satiety signal doesn't get through to the place in your brain that says, I'm good, I have enough energy. And so you have then a lot of sugar cravings, a lot of cravings for higher fat foods. And if you let people self-select the type of foods that they eat, they'll typically choose the really energy-dense foods in the group that has this variation.
Right. Another one that's actually related to BMI and fat gain is the ApoA2 gene. ApoA2 codes for a protein called apolipoprotein A2. It's the second most abundant protein that's found in our HDL cholesterol. So it's a good cholesterol. It's the cholesterol that actually takes, it's the protein that takes the cholesterol from the arteries and deposits it back into the liver for recirculation. So we know that it's not only has anti-atherosclerotic properties, it's anti-inflammatory and antioxidant as well. And there's a number of studies that look at a TC variant, and this RS number, you see, that just tells you the exact spot in the genome where that variant is found. So it's an RS5082. So that TC SNP is involved in physiological responses to fat intake, so the fat that people consume. CC genotypes are much more likely to gain weight when they eat a diet that's high in saturated fat. as compared to either TT or TC genotype. And this is, I'm not sure if it shows up very well, but if you look at the left-hand side, you can see that the open circled one is, maybe, yeah, Dallas can point here. The CC, so the x-axis is saturated fat intake, and the y-axis is BMI, or body mass index. And if you have a CC variant, for low saturated fat intake, much lower BMI, but it goes up very quickly as soon as you increase your saturated fat intake to much higher BMI, and it surpasses that TT or TC variant. And in the graph on the right, again, BMI goes up and down on the y-axis, and you can see that the light gray bars are the TT or TC, and the dark gray bars are the CC variant. Low fat on the left, high fat in the two bars on the right. So if you have a low-fat diet and you have a CC variant, you probably have a low body mass index. But as soon as you have a higher fat intake, and in this case greater than 22 grams, and you have a CC variant, you're going to have a much higher likelihood of gaining weight and being obese from that. And so how does it actually do this? It does this through ghrelin. It modulates our ghrelin release. Now, ghrelin is a hormone that is produced by our stomach and our pancreas, and it stimulates hunger. Levels actually increase before meals, and after we eat, it decreases. And it acts in contrast to leptin. Now, this is, again, looks at ghrelin levels based on ApoA2 genotypes, and the level of ghrelin goes on the y-axis, and you can see on the left is the CC genotype. A, and the one on the light gray bar is low, then middle, then high fat intake. Game over. Is high fat intake, and you can see that the level of ghrelin goes up much higher in those people than in the other genotype when they eat high saturated fat. So in this case, if you have a CC genotype, you don't do well with high amounts of saturated fat, and that becomes very relevant when we talk about a paleo lifestyle where saturated fat is considered very, healthy. We have to think about that.
Well, and you could use some of this information, if you had this information about yourself, you could use that to make more educated choices and say, if I happen to have a CC variant in this particular gene, then I'm not going to do really well with choosing foods that are really, really rich in saturated fat and making that a primary part of my diet if I'm trying to control my body weight. If that satiety signaling has been abnormal for me and I'm having difficulty kind of maintaining a healthy body weight, and of course we know all the health correlates with obesity. So it's a good example of how you could take some of that information and change what you do, understanding how your body, because of your genetic makeup, is going to respond to the environment, the soil that you live in.
Mm-hmm. So our time is up. There are lots of other examples, and based on our questions, I'm happy.
When they say game over, they mean game over. You're done. Like the hook is coming off the stage here. We'll share this. That's fine.
Any questions?
Hi. Do you have a lab that you recommend? Not an actual test that looks at all the different variants, I guess.
Mm-hmm. Yeah, we do. So one of the things that Dallas and I are going to start offering is this testing for people so they can start looking at those individual variants. And I am the medical director of a lab in Toronto that will do all of this testing that you can get through. And we can look at all of these different SNPs and variants. Absolutely. Yes, we are doing it. Yeah. Yeah, if you wanted specifics of what you really want to, sure, we can talk about that later. Absolutely.
Hi. If you have the CC variant, what should you do for your protein?
So that's a good question. And understand that these are single genes, they're influential genes, but they are not the whole story. So you have to kind of contextualize some of this information. You shouldn't build your whole life and your whole dietary choices around this. But it's a fair question. We're not saying that saturated fat is harmful for people who have the CC variant. What we're saying is it changes the way ghrelin signaling drives behavior, drives hunger and appetite and satiety. So in that case, for a CC variant, in a simplified way, an example would be, hey, just choose leaner cuts of meat that aren't going to be so rich in saturated fat. And that's not avoid saturated fat. That is just don't start your day with a pound of bacon, right? So again, you're looking for concepts that are still very much within that ancestral framework. You're just not going to extremes to kind of really pick out and emphasize some of those very saturated fat-rich foods if body weight regulation is something you struggle with. Does that make sense?
So 23andMe got a cease and desist basically from the FDA. How are you guys planning on how to get around that already? And also advice for keeping people from completely going off the deep end with this stuff, because obviously genetics is way more complicated than the layperson is going to understand.
I'll take part 2 and then I'll let Helen take part 1. How do you keep people from going off the deep end? I think the thing that we try to kind of present is some examples of some of these things and how they influence or how they could influence the choices you make. But even just to my kind of precursor to my response to the last question is, this is just one small piece of a very, very large and complex puzzle. And we don't want people to take one small bit of information and totally reconstruct their life around it. I think the thing that is challenging for those of us who have done kind of the whole genome sequence, who have all of the raw data from something like 23andMe, is not going off the deep end, because there's so much stuff that's so interesting and so potentially influential, but the reality is that so little of it is really well understood in terms of real life clinical relevance. So honestly, from a individual perspective, I think finding somebody who's really kind of fluent in genetics and knows how to interpret that stuff is really at this time going to be your best bet until we have so much more data on clinical real life relevance, because there's lots of things you can look at that modify certain kind of responses to our environment that are completely irrelevant in real life. And I think knowing which is which, partly is experiential, but more importantly is reliant on having there be a robust data set to say these 27 studies all are consistent with how this happens in real life. And so at this point, I don't think there's an easy way for individuals to look at their whole genome and interpret what that means for real life. One of the things that Helen and I are doing is we're kind of taking mostly Helen's expertise with genetics and going to be selecting out some of the SNPs that have the most robust data to back them up to be able to make actionable lifestyle recommendations based on the things we like. There's really good data here, and not putting forth speculative recommendations based on one study or one association. That's kind of part 2.
And part 1 was, how do you get around sort of the FDA shutting 23andMe down? Well, the FDA's issue with 23andMe was twofold. One was it wanted to make sure that their data was done properly, and they actually do a good job. They use an outside lab to actually do their testing, and if you want, I can tell you what that is later. But and they do a good job of that. The other issue was that they were doing direct-to-consumer marketing, and the FDA felt that consumers weren't quite ready to hear this information. So they didn't like the way that 23andMe interpreted the data for consumers. So in the I don't know if any of you ever got some of those original reports, but you would get something that said you have a 20% risk of rheumatoid arthritis, or you have a 2-fold higher risk of cardiovascular disease. And it wouldn't tell you what it was based on. It wouldn't give you any of that other information. You're just handing this out to people without having any idea what to do about it. Okay, now I have a 2-fold higher risk for cardiovascular disease. What do I do? So they're still allowed to give the raw data. They can still say you have an AT change in this area of your genome, but they can't say this puts you at higher risk for something. And so that's the thing is, you are allowed to do that if you do it through a healthcare practitioner or someone that's going to help you interpret that information. And I mean, just one example is I'm one of very, 0.00001% of the population. I've actually had my entire genome sequenced, and all 3 billion base pairs of it. That's a lot of information. And even though I'm a geneticist, I don't have any clue of where to start delving into all of that information. So we're not at a point yet where it makes sense to sequence everybody because we don't know what to do with it. But we will, it's important to take the ones we do know a lot about and test for those so we can actually make changes based on good, robust knowledge.
This will be the last question, sorry. Okay. Can you comment on some of the, right here.
Oh, there you are, sorry.
Can you comment on some of the new information in regards to epigenetics and how the environment's affecting gene expression?
Absolutely. I mean, it's critical. So here's a good example. The top one of the list here, CYP1A2, stands for cytochrome P450 1A2, which is involved in caffeine metabolism. So we know that you have a genetic change where you're a fast metabolizer or a slow metabolizer. So you can either drink that espresso at 10 o'clock at night, be asleep in half an hour, or you're going to be up all night, right? I'm a slow metabolizer, but I love my coffee, probably because it has a much bigger effect on me. Now, we get into the whole epigenetic concept of nutrigenomics. We know that broccoli seed extract upregulates the expression of the CYP1A2 gene. So now I can drink my coffee and eat broccoli at the same time, and I can handle more coffee or more caffeine. The opposite, turmeric, or the active ingredient in curcumin, actually downregulates the CYP1A2 gene. So if you have curcumin with your coffee, you're going to have the effects stay around in your body a lot longer. So that's an example of an epigenetic, environmental change on our gene expression. It's very important. Epigenetics are as important as our sequence, but you have to look at both. And does that help?